All right, very excited to introduce our first speaker. She is actually our first grant recipient. Just like this event, we thought everything would have gotten finished quicker than it did, but you know, the pandemic. So please welcome to give us some updates on the BioGENE study, Dr. Caitlin Hudac from the University of Alabama.
Thank you, everyone. I'm so excited to be here, and I see so many faces. I don't recognize all of you right away, and then I start to see your kitchens and your living rooms in my head, and then it keys in. So if you don't see that immediate jolt of clarity, feel free to be like, hey, remember me? And I'll key in. So I'm really excited. We had such an amazing year doing this study. The pandemic finally hit a point where we felt comfortable doing it, so I'm really excited to give you some updates and some future directions. But just like Liz, I thought maybe it'd be helpful to actually start by telling you a little bit about where I come from. So many of you might not know, I'm actually from Mars, Pennsylvania. Go Steelers. So I was kind of born and raised by a psychologist and swore, I'm never doing that. I'm not going to go into psychology. I'm doing the complete opposite. I'm going to be a hand surgeon. I had lots of ideas. But then I went to college in Chicago here. There we go. And what do you know? I became a psychologist. So I actually, after college, I decided I wanted to figure out what do I want to do with this? And so I actually worked really closely in a residential facility down on the south side that works with kiddos that can't quite make it in day school and need to be in a residential setting. And I worked day in, day out. We did all sorts of activities, and I realized I wanted to know about what was making their little brains work. And so I actually found myself looking for some research opportunities. And so I moved back to Pittsburgh and did some work using fMRI in Pittsburgh. And then we actually moved to Connecticut as well. And so my first kind of research job was in fMRI, which uses the big magnet. Some of you might have experienced it. So I would work with kids from birth through adulthood, getting them through a scanner and kind of having them sit and listen and watch movies that we played. And so then I took that and went to grad school where I learned this really cool thing about EEG, electroencephalography. So I did a lot of work with infants. So I kind of had this autism experience, and then I had this experience working with kiddos that can't talk to me. And so I had kind of that sense of what we could do in lots of pre-verbal or nonverbal populations. And so when I moved to Seattle, for my postdoc experience, I got really involved in the genetic work that we were doing at the University of Washington. Some of you might have heard or participated in the TIGER study at the University of Washington, and that's actually where I became involved in GRIN2B, and I met my first kiddos with GRIN2B. And I, on a whim, as I'm trying to sort out what comes next and wanting to become a professor someplace, I applied for the GRIN2B grant award and was very, very excited to have received it because that was able to really fund the work I wanted to do in seeing as many kids with GRIN2B as possible. So I am actually currently finishing my first professorship at the University of Alabama, and in the next week or two, moving to South Carolina, where I will be doing more of this work. And I'll kind of key you into some of that at the very end as well. So I've kind of lived all around the country, and as a preview, I've seen a lot of the country in the past year. I also just want to acknowledge that the University of South Carolina is land that was part of the Cherokee East people. I want to acknowledge the land that we will be working on. So I am what we call a developmental cognitive neuroscientist, so psychology background, but I really focus on development, so how we grow, and specifically how our brain grows and how it processes information in the world. And I'm really interested in this lifespan perspective, including understanding how our genes that are really nothing that we can control are going to influence how we think about the world. I have a lot of hopes that if we can kind of identify early periods of risk, potentially looking at these brain markers, that we might be able to change trajectories and really understand when and what kinds of clinical trials, what kind of behavioral testing, what interventions we might need to do to help boost some of these developmental processes. So when it comes to kind of thinking through how I fit into this kind of clinical trial model, for those of you that aren't like super familiar with how kind of this world works, we start with understanding these basic science mechanisms. And this can be looking at animal models or like what we do when looking at the brain in humans. We have to then translate it to what we see behaviorally. So how is what we're seeing kind of in this basic period actually reflecting back to what a child is doing on a day-to-day? Then we develop interventions, and we test those interventions, see how well they're doing, and we'll hear a lot about that next, I hope. I'm excited. And then we have to implement it. So all of these are really important pieces of this kind of cycle, and we kind of see ourselves sitting at the beginning part of this, right? So we're really trying to understand that link back so that we can inform those intervention treatments. So if I could say what would make me feel like I accomplished my career goal, it would be to complete this circle and really get us to the point where we're not only understanding what treatments we should be doing, but how they work and why they work and what kids need what treatments. So that's really, but give me some time. That's a long time coming, but that's really where we're hoping to go. So today I want to kind of talk a little bit about what biomarkers are specifically and why I am really excited about them in GRIN2B and talk a little bit about what a lot of you got to experience as well. So I want to highlight all of our GRIN2B Warriors that participated. And as an early shout out, we also are here to do research. So if anyone has a child who's here and hasn't signed up, I'll key you into how to do that at the end. And then I want to talk a little bit about what our next steps are. So the critical problem is that for all of our children that have a neurodevelopmental disorder like autism or have a genetic diagnosis like GRIN2B, we tend to see that there's widespread what we call heterogeneity. So each kid can look a little bit different. So if you have this kind of variability in your population, let's say you just try to randomly implement different treatments, maybe these 3 different treatments. Well, if the treatment isn't quite meeting what that child needs, then you're going to have variable treatment success. So really what the kind of role is and our job is to try to understand what are these subgroups. So for instance, for autism, we want to try to separate into different patterns and understand, well, okay, so then if we implement those 3 treatments, we want to have 100% success if possible. And our thoughts is that maybe one of these subgroups is going to be related to GRIN2B. And so we're really trying to understand what is happening uniquely to GRIN2B. So what are biomarkers? Well, it's complicated because people used this to refer to different things. So a biomarker is a biological indicator of some sort of clinical disorder. So these could be, so just thinking of autism, for example, we diagnose autism as a behavior, right? So that's not necessarily a biological indicator. So we have to look at a different level of our little pyramid here and maybe use some of our brain techniques. Work in concussion biomarkers, for instance, uses a lot of looking at blood platelets and things like that. So you can use really any sort of biological specimen to try to understand this. And then what our role as scientists to kind of make sure that before we push things up that little circle into implementation of treatment is to understand how is this biomarker working? And is it really separating out kiddos that have, for instance, autism or GRIN2B from kiddos that don't? So we do a lot of this work, and I'm not going to get into the nitty gritty, but this is what we are trying to do with the BioGENE study. So there is a lot, go ahead, Michelle. Sorry, thank you. That's neurotypical. So in just this example, this is kind of thinking about autism versus kiddos that do not have autism, so kind of the absence of the diagnosis specifically. So in a GRIN2B example, it would be GRIN2B potentially compared to maybe a neurodevelopmental group, or when we can kind of chat, what your comparison group is can also vary. Good question. Thank you. So there actually are some really big or consortiums that are working on developing biomarkers for autism. That's great. It's very exciting. they're using EEG, they're using eye tracking, but they're not necessarily going to be helpful for all populations. So most of these large consortiums like the ABCCT study and the EU AIM study actually have IQ cutoffs and verbal functioning level cutoffs, and that doesn't necessarily fit all of our populations. So if you have a population that has communication difficulties or sensory sensitivities, then these might not necessarily be the right first step. So what we're trying to do is take some of those principles and move from having it be kind of having a kid sit in an EEG net and playing a game and look to see, well, can we use things that are not task-based, so not playing a game, but just sitting, listening, looking. And what I like to call, I think a lot of you heard, chill and still, right? So kind of getting into that state to understand those brain processes. The other piece that's really unique for us is the ability to travel, right? So a lot of these studies are at major universities in big cities. If you're in a rural population or far away from one of these cities, then you might not necessarily even have access to these research pieces. The other thing that I just want to mention, and I've been thinking about a lot when working through kind of what we're seeing in GRIN2B, is a lot of these consortiums are autism focused. And one of the ongoing questions, I'm just going to kind of leave it out there to hang for now, but is autism really going to be a focus for this community? And I think that that's an ongoing question. I already see some head shaking, so maybe it's less unknown, but we're building from autism work, so that'll be our kind of framework for today at least. All right, so for GRIN2B, when I think about how this pyramid works, so we know that GRIN2B has a very particular genetic role that I'm not going to try to explain in depth. I'm going to leave that to other people. But we can kind of think through, so GRIN2B is a gene that's going to encode for NMDA receptors, and then those are going to kind of have these cascading effects, right? So the proteins that you're able to produce are going to be this cascading effect. But when we kind of think through our kiddos, we know that there are these kind of common diagnoses, and Liz mentioned several of them, right, when talking about Lucy. So we see a lot of language and learning delays, in particular, hypotonia, other kind of motor delays, smaller head sizes, and then some level of kind of autism or autistic traits potentially. So there's been some new papers, and I think that my references are getting cut off, but I can send the slides around. But this year we had a couple new papers come out, one looking at GRIN2B brains, and specifically found that there's these brain folds here in what's called the hippocampus, which is our memory hub, and we're seeing what's called atypical gyrification. So the way that the brain folds in the hippocampus specifically is atypical. There was another paper that came out looking at zebrafish, and so they have a zebrafish model with a knockdown specific to GRIN2B, and they're looking at how that knockdown then relates to social behavior. So kind of thinking more of if we change the gene, how does that reflect back up to our social behaviors? And they found that the GRIN2B zebrafish were less socially engaged. So this is all really cool. As a scientist who's really kind of sitting aside of that clinical and that basic science piece, it's helping me to kind of start to think through what's going on. And this led us to kind of think through, well, what are we seeing in our kids? So we have a paper that we're still working on and adding a lot of the BioGENE data to. And really what we're seeing from parents' reports is very much related to this autism question, where we're not necessarily seeing classic autism traits. So our kiddos are socially interested, they're motivated, they want to approach. And we're not seeing those same kind of motor stereopathies and restricted interests that we always see in autism. And we're seeing these really, really great strengths in that social piece in particular. And this matches what we see from our clinicians too. So I worked with our clinical team at Washington to go through all of our kind of coded clinical assessments like the ADOS and really look to see what responses we're seeing. And there might be some unusual behaviors. So sometimes the social approach is really positive, but the kiddos don't know quite how to engage in that situation. So we're having some mismatch, but really it's not because there is this lack of social interest. This is just highlighted by some of our other work. So this is just a parent report a lot of you filled out called the Social Responsiveness Scale. So everything kind of like where that arrow is and above, it would indicate having social problems. And when you look at the overall score, you can kind of see GRIN2B here in the green is actually very similar to other genetic mutations, as well as autism, both with and without co-occurring intellectual disabilities. But when we look at the motivation part of this measure, it's showing us that there is not that social problem in GRIN2B. So it's more evidence really suggesting that there's something really, really great and exciting about how our kids want to engage with other people that does not resemble autism. So just kind of summarize here, you can kind of see like we've got like some great coverage, even in these little studies that I featured, but we're really missing this kind of hole here in the middle. So that's really where the BioGENE study comes in, and it's meant to try to be a bridge between these low-level features related to our genetics, our proteins, how all of our body is kind of creating these networks, and then linking it back to what we're seeing at the diagnostic level, understanding how that relates to behaviors. So this is really where we're going to target with our biomarkers. So the BioGENE Study, which is actually an abbreviation that I weirdly came up with. So it's the Biomarkers of Genetic Etiology of Neurodevelopmental Disorders. We have a new website, so for our move to South Carolina. So we've been updating this, and actually there's some updates as of last night even. And we're really building from the work that I did as a postdoc, working with the tiger and the zebra studies. So remember, I'm a developmental cognitive neuroscientist. I work on the brain, really. That's my focus point. So we're starting with our brain markers, and I'm really hoping to kind of keep connecting back up to clinical symptoms as well. So right now, we're really just focusing on a couple of genes, and today I'll talk to you about what we're finding in GRIN2B. So for those of you who don't know what electroencephalography is, or EEG, this is just kind of a quick little overview. So if we have a part of the brain that's active, it's going to send out electric signals, and it sends them out in a kind of interesting pattern. So we see just negative signal, and we see positive signal. So what we're really trying to do is we put these caps on our kiddos, and then we look to say, okay, at every millisecond, what are we seeing at these different recording sites? And that's telling us about how different parts of the brain are engaged and active. Now, EEG is really cool, but it's also going to pick up on other sources of electricity. So your body has other, like just breathing creates electricity, the lights create electricity, movement creates electricity, like if you smile or laugh, that's going to create electricity too. So part of our job as scientists is to say, okay, what is just these other artifacts, and what really is the brain process? So here's just an example of how quickly the net could go on. So this is from work that I did in for my PhD. So you can see seconds, right? So we use this system specifically. It's expensive, but because we can do this, the net just goes on super quickly. We're able to record at any age point. So we work with infants all the way through adulthood. And we have over 100 electrodes we can record from. So let's say even if a kiddo is picking at a couple electrodes, it's okay because we've got other ones that we can use. So it's a great way of kind of quickly getting a lot of data. Now, those of you that are parents that have had to go to the hospital and sit with the gel, very different, right? So when we talk research EEG, we do this on purpose because we know that this is easier. Now, a lot of you are very curious if we can tell you kind of clinical results. We don't necessarily have that specificity because what you get by having the gel electrodes is having more, we're concerned about some of the movement pieces that you don't get with the gel. So the gel is really great for clinical pieces, but we're able to still get great cognitive data. So back in Washington, I started kind of thinking about what does this look like to travel. So we had a couple of road trips that we traveled where we had a portable system that we would bring and set up. And so I got really used to kind of what that looks like. And it's a wonderful opportunity. So I think some of you participated actually in the Tiger study very, very early on where we came out with our system and set up. It actually can be less expensive from a research perspective. And one of the best pieces of this is that the kiddo is in their home. So it's where they're comfortable. Their favorite toys are there. If they need to take a break and go hang out in their room for a little bit, we can take a break. So it really just kind of creates an interesting environment where we can put everyone more at ease. There's some tricky pieces to it. So each environment is different, right? So we have our setup right now in the hotel, up in my room actually, ready to go for anyone who's interested. And that is a different environment than, let's say, someone's kitchen or someone's bedroom or our laboratory environment. So we have I've got lots of ideas and happy to talk about the science of how we try to control for that. And it really does require having some specialty skills. So some of you might have, I don't think that we had too many technical problems with any of our GRIN2B kiddos, but if you're missing a cord or you forget your potassium chloride at someone's house, and can't make your electrolyte solution, some of these can be issues that we can face. And it does take some amount of time to set it up. So when we set up our system, it's nice to, like this, to have meetings where we can then test multiple kiddos at once. So this is what we did this fall. We figured COVID was pretty much at a good lull, I guess we'll say. And I masked up and I drove my car to Washington to spend the summer with friends. And then I drove around the country by myself with our EEG system. So altogether, I logged over 15,000 miles in my little Honda Accord. It was just over 3 months, so 92 days of testing. And we ended up seeing 55 kiddos and one parent, actually. So we did this with both GRIN2B and another gene, SCN2A. So one of the other pieces I just want to mention is that we're not done. There was a lot of people we missed. So, and some of you are here today, and we're hoping and planning, and Rebecca and I have already been kind of thinking through how we're going to get to see everyone. So we, like I mentioned, it was incredibly successful. We didn't necessarily get a net on everyone. There was one kiddo who it wasn't going to work, and that's okay, because our kiddos have sensory sensitivities. We tried a couple times and it wasn't in the cards that day. And that's totally fine, right? It's just great to kind of get there and get to meet everyone and see. But one of the things that was, I think, really a great thing to highlight is that we had a lot of rural families, which I don't think I wrote on the slide, but the fact that we were able to access people that don't always necessarily have access to medical centers, and really almost absolutely everyone who had a net on their head was able to, actually I take that back, everyone who had a net on their head completed at least part of a task. So following data cleaning in this first pass, I'm going to talk today about some of the results from 27 of our kiddos. Oh, and this study really doesn't, I did a lot of driving, but I wanted to really feature all of the work that was done. So unlike Phil, I'm not necessarily a video designer, but I put together a little hype video as well. I don't think the sound's working, though. Does anyone know how to do the sound?
Passing hours, pinching all the pollen from the cotton bean flowers.
And sometimes I fell asleep. So I'm really just proud of how well all the kiddos did. It was such a team effort. A lot of you were very actively involved. I don't know, I should have counted the number of M&Ms and veggie straws that were used and pancake breakfast. And we definitely had some hurdles. So we had a couple of kiddos that were really, really anxious about it. So what you see here is that I put on a net. So I was wearing a net while she was also doing her session. And we made it through gloriously. So I also want to just kind of highlight Natalie, who is here. So she actually has done this before, and she had some initial hesitations. We thought, I think there is sound. We were telling her.
Happy, 1, 2, 3, happy. Oh, that's evolved.
So don't let those tears fool you, because this is how we ended the session, right? So we get to this great spot at some point. And actually, after the net came off, we still had paperwork to do. And Natalie and I were just hanging out, and she asked to put it on multiple times. So clearly, she still was a happy camper at the end after I initially brought tears, which always breaks my heart. So the question is, what biomarkers did we want to target? Right, so clearly we've executed this. And so I was kind of thinking about this list that we know about GRIN2B, and we really kind of wanted to kind of highlight this piece. So thinking if we can connect what we might know about some atypical parts of the hippocampus that are related to memory and to learning, to what we know about with language delay and learning issues as well. So we specifically wanted to ask about two things. One, are there these basic skills? So what are our kids paying attention to? And then how are they learning about that information? So why do we care about attention? Well, it's actually what we call a low-level part. So what we look at and what we decide to attend to is going to dictate what information we are gathering inside our head. So that is really what we experience in our life every single moment of the day. So one of the pieces that we know in psychology is that when something new happens, we have kind of what we call a pop-out effect. So when you see something new or hear something new, you're going to orient towards it. So for instance, can anyone see what's a little different about this picture? It's that of all of these stuffed animals, there's actually a real dog in the middle of that. So now that you know that, it actually gives you this context for learning. So if I were to kind of familiarize you with other pictures similar to this, perhaps you could figure out where those pop-outs are as well. So it's one of the classic ways that we as humans learn. As we are developing this information, then we're able to kind of like diminish our response. So what we expect to see is what's called habituation. So as we're learning, we're seeing this response, this pop-out effect specifically, get smaller and smaller. Does that make sense? So we actually use auditory tasks because we know a lot of our kids aren't going to want to watch the screen, and this has been something that's worked very, very well. So we have two experiments that I'm going to walk through. So what we do is we have them watch a video or their iPad, or they can look at their thumbs. Whatever they want to be looking at visually is totally fine. Again, the goal is still and chill. And then we play these sounds. And one of the things that you can kind of see is that we've got the one tone repeated over and over and over again, and that gives us these opportunities to look at kind of a repeating sound. And then we specifically look at, well, what happens when the tone changes a little? Or what if it's a totally different sound? And actually, could you make sure the sound's on again? So I'll play a little clip of this session. You get the point. So they're watching this video, they're hearing these sounds over and over and over again. And we've actually published on this before. We know what this looks like in autism. We have a ton of kids on this specific task. So it's great for understanding how is GRIN2B different and unique. I also wanted to try something a little different given the fact that we see this kind of learning and language component in GRIN2B. So we also developed a social oddball task where instead of having it be tones, we're using speech syllables. So we're looking to see, are we seeing a difference whenever the speech syllable is different? So having a repeated ba and having it be da, or also how that relates to that novelty effect again. So here's another example.
La, 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 la.
So it's subtle. Sometimes you don't even, as adults, it's hard for us to tell. But there's just one consonant difference, and so our brain should be able to pick up on that. So what we do with our EEGs, we take that ongoing electric signal, and we're plotting it. So where that little vertical line is when the sound occurs. So sound happens, and then we're measuring the electricity after that. And we can look at a couple different things, but today I'm going to focus on what we call the P3A. So this is just one part of the signal, and we think it's related to orienting. So when I was talking about that pop-out effect, this is related to that pop-out. So kind of directing your attention towards that stimulus. So what we expect to see is that we're going to focus on those novel sounds versus those repeated sounds. So this is novel sounds versus the tones in the first experiment, which is really just this broad attention task. We expect that red line to be bigger. And what we're seeing in GRIN2B is just that. So we're seeing this kind of, our prediction is met. So we're seeing that a bigger response for those novel sounds relative to our frequent tones, which is really exciting. But my lab loves to think about this learning piece and thinking about dynamics and time. So what we know in kind of neurotypical individuals is that red response actually has a unique shape over time. And in neurotypical kiddos, we see it fall pretty quickly. Remember, I was talking about habituation. So this is our learning response here. Well, what we actually found in this paper for autism is that it does not fall as quickly. So in autism, the kiddos are staying attentive to those sounds, and that can be problematic because then you aren't necessarily redirecting your resources. So you're using a lot of your cognitive resources to focus on the pop-out effect instead of moving on and thinking about something else. So what do we see in GRIN2B? Any guesses? Oh, yeah, and this is also linked to sensory sensitivities. We actually see a very similar pattern to our control kids. So we aren't necessarily seeing that autistic hyper kind of responsivity that's kind of enhanced and hanging on. We're seeing that this is fading at a very similar rate to our neurotypical controls, which was interesting for sure. So switching to the speech task. So this is what we'd expect to see if you're going to have differentiation, right? So having a bigger response to those novel sounds versus speech syllables. And we actually did not see any differentiation at all between sounds and a repeated speech token. And so I was like, oh, it was kind of interesting. I'm still not entirely certain what to make of this. And again, we don't have comparison data on this task yet. We're going to be working on that. But so I was like, well, maybe the learning patterns will make sense. And so we plotted those. And you see how the lines are very similar. So that's why we're not seeing any difference between the two. But we're seeing this really interesting. So we're seeing the signal decrease initially, and then we're seeing it increase towards the end. So really, what we need to be doing is kind of unpacking that a little bit. So why are we seeing this modulation of the signal? I won't really know more about that until after we see some additional kids, neurotypical controls, maybe an autistic sample as well. It could be describing, modulating saliency, so just interest. We also did this task last. And some of you might know from your kiddos' experience that they do get a little tired. This is about after 12 or so minutes, right? So it could just be a fatigue effect. So that's something that we'll need to test as well. So to be able to do that, we're really planning on kind of continuing our data collection. So we have some families signed up today. So this is Rebecca Revilla, who's my graduate student. And Rebecca, if you could wave or stand. So if you are here with your kiddo or your kiddo is local and you're interested, we have everything set up. Check in with Rebecca to figure out the schedule and when you want to participate. We'd love to have anyone sign up. We're really open to all ages. As long as you have a disrupted GRIN2B mutation, anyone is really eligible to participate. So then the last other thing that I want to just kind of point out is that one of the reasons why we're moving to South Carolina is that I have a lot of future colleagues that are really working on this lower end of our pyramid, and they are really excited about GRIN2B. They've worked a lot in other autistic genes, but they haven't necessarily worked in GRIN2B, and now they're really fired up that we're coming and they want to work with us. So I kind of listed out some of their specialties, and I'll share the slides. If anything kind of comes to mind that where we all want to work together, any of the other researchers in the room or our family members have specific questions, we're really excited to team up with them to kind of link what we know at the human side with what they're working with cells and animal models. So I just want to wrap up, and I'm sorry if I'm over time. I wasn't timing myself, Liz. So we really do see, I'm really fired up about the opportunities that EEG offers, but I also recognize that we're looking at that one scope, right? So I kind of put ourselves in the middle of that pyramid on purpose. We need to be able to link above, we need to be able to link below, and our expertise isn't going to change, so we really need to connect with researchers that are interested in those pieces. And there might be other things but there might be other things that we can address. So I have experience in fMRI, I have experience in eye tracking. We're going to be doing some eye tracking, EEG, and heart rate pieces all together at once when we get to South Carolina. But what we need to know from the families is, what are those questions? What do you want to know? What is out there that we can help you sort out? The other piece is that, I think it's exciting. I was jotting notes as Liz was talking about how big this organization has grown and how many families there are. That is so incredible, and the fact that we're going to hear about clinical trials next it's beyond exciting. It really, really is. And so we need to understand kind of, where do we go? And also, how do we fit in with existing consortiums that are doing some of this work, but maybe from an autism focus? So if autism isn't a focus for this group, how can we potentially maybe capitalize on what they are doing in a way that works for GRIN2B? And then lastly, a big passion project for us is that we really want to make sure that research is inclusive. So this includes being able to go to families that can't travel to you. And that might be for a number of reasons. Taking off of work, having a kiddo that really isn't travel ready all the time, or just not having the time. So we want to make sure that we can bring that to you. We want to work with kiddos that are historically excluded from EEG work. So we have done a great job in Alabama of working with our kiddos that have Afro-textured hair, braids, dreads, wigs. We know how to do the EEG with them, and we want to include more of our individuals that are historically underrepresented. And then we need to go international. And I know that comes with money, and so we're getting there. So that's really the next step. But we really want to be not only focusing on our United States groups, we really want to understand what's going on worldwide. All right. Sorry if I'm over in time. I just got really excited. So thank you all for listening.
If there's any questions, we'll take a couple minutes if anybody does have any questions for Dr. Hudac. Yeah. Actually,
Hi, my name is Michelle Gauthier. I have a son, 4-year-old son, with GRIN2B, and we're in Ottawa, Canada. I don't know about others here, but I didn't quite understand the results you got about the speech and how it relates. I know you're running out of time, so you didn't have a lot of time to go through that. Can you just explain that a little bit more and what you hope to explore during your second phase of the study?
I knew you were going to catch that. So to be honest, I don't entirely understand it myself just yet. So part of it is because we are only looking at the GRIN2B kiddos, and that really surprised me. So that was something where we expected, because they have this language delays, I expected, yes, there might be some differences in how they represent the ba versus the da. But what I was showcasing here is the pop-out effect of those novel sounds versus a speech token. And actually, last night I was trying to sort this out because I was like, I needed a better answer. Or I'm not seeing any difference between these novel sounds, the repeated speech token, and then the deviant speech token. So we're actually seeing very similar brain responses to all. So it's kind of like the brain is saying, I heard something, but it's not differentiating that something is different occasionally. And that's surprising. But I don't know, again, because we haven't looked at this in other groups. I will say I did look at the SCN2A kiddos, and they are showing a difference on the speech task, but they're not on experiment 1. So it's kind of like GRIN2B and SCN2A are crossing over right now. But I don't know what's meaningful about that, but I will continue to keep you posted. And that's really kind of our first mission once we finally get settled in South Carolina. Mm-hmm. Mm-hmm. So for those of you who couldn't hear, sorry, I probably should have passed the mic back to you, Michelle. Michelle was talking about having, needing that kind of visual cue with the auditory cue to differentiate between sounds. And that's really exciting and good to know, because there's a part of our brain, and I could wax poetic about this, that is specifically processing how visual and auditory information are linked. And part of that is actually in the hippocampus. So it could be the case that maybe we need to be thinking about the specificity of sensory processing more broadly. We picked the auditory domain because we know our kids can do it, right? And it's very successful. But maybe what we need to be doing is thinking about adding in a visual component and seeing if we're seeing the same sort of attention signals, and if language is improved maybe when you have that the visual cue as well. So that's a great next step for us too. I have a feeling I'm probably, oh, God.
This is going to have to be the last one. I apologize, everybody, just because we do have more things to get to, but go ahead.
Thanks, Phil. Hi, I'm Gillian, and I'm from the Netherlands. And perhaps I'm totally wrong, but I thought these graphs you had were dealing with automation. You see a task, and you do it quicker and quicker. You automate it. I was quite surprised that our kiddos do it almost the same as neurotypical kids, because my experience is that you have to practice for like forever before your child masters a task and also keeps it and doesn't forget it again quite quickly. How does that relate?
It's all good.
That's a great question. So remember, this is it's what we call a passive task. So it's not necessarily linked to an expectation of behavior. So there's a part of our brain that is doing this attention signaling that is going to happen whether or not, even if you were to sit down with an EEG net and you were ignoring the sounds and you're thinking of your grocery list and what you have to do the next day, there's a part of your brain that's still picking up on the sounds. So even if you're kind of task distracted, we see this signal, which is, I think, an evidence of the fact that this is a robust, what I was calling a low-level marker. So it underlies the other pieces. Now, if you were thinking about your grocery list, your responses are going to be diminished. But we're really focusing, because everyone's in different contexts, we're focusing on the condition difference. So whether or not you're thinking your grocery list or a kiddo is, the signal's becoming more automated in our kiddos, it's really about the relative effect between the two sounds that we focus on. So it's unlikely that one is going to be impacted by distractions or automaticity and the other isn't. So this gets into the complicated brain scientist part, but I feel very confident that these are real underlying signals. I don't necessarily know how they would relate to explicit behaviors in the moment. So if you were to ask your kid after I left, oh, what did you hear? I don't know that our kiddos would remember or have necessarily a fine-grained understanding of what they heard. We're kind of banking on the fact that we get the low-level part and that later we can understand how that links to higher-level pieces. Okay, thank you for letting me answer some questions.
Thank you, Dr. Hudac.